Did you know that all cells in your body can produce extracellular vesicles, or EVs? These tiny particles provide snapshots of a cell's state at the time they were released, making them a goldmine of biological information. Because all cells produce them, EVs are found throughout the body, and one of the best ways to access them is through biofluids. Biofluids are liquids found in the body or secreted or excreted from it. Things like sweat, cerebrospinal fluid or CSF, milk, saliva, and lymph fluid. Some biofluids, like ascites, are linked to underlying illness or injury. Just as all cells produce EVs, all biofluids contain them. The types of EVs in a particular biofluid depend on where and how the fluid is formed. CSF is rich in brain-derived EVs, while urine contains EVs from organs like the kidneys, bladder, and prostate. This makes biofluids an exciting avenue for biomarker discovery. They allow researchers and clinicians to gain insights into specific tissues without invasive procedures. Obtaining a urine sample is much easier and safer than a kidney biopsy. Biofluid EVs are being used in the hunt for biomarkers for a multitude of conditions, from cancer to gout. But turning this potential into reality isn't easy. Challenges exist at every stage, from optimizing biofluid collection through to identifying a readout that provides clinically relevant information about any underlying pathophysiology. Let's start with collection. Different biofluids have different characteristics. For example, urine is generally a dilute, high-volume biofluid with non-EV contaminants including cells and microorganisms. A specific consideration is the presence of uromodulin, a glycoprotein that polymerizes, particularly at low temperatures and after sample concentration, increasing sample viscosity and potentially trapping EVs. In contrast, blood is a highly cellular biofluid that must be processed into cell-free plasma before EV isolation. Plasma has a high protein content and is often limited in the volume available. A key consideration is platelets, which are a major source of blood EVs and may continue to produce them ex vivo, changing the composition of the sample after collection. Factors like quick processing can be helpful in minimizing this contamination. Other biofluids all have their own unique features to consider. Saliva is highly viscous, CSF is limited in both volume and EV concentration, and milk is a complex colloid containing fat globules and casein micelles that complicate EV purification. The EV field is still in the process of cataloguing these sources of variation and standardizing methodologies for reproducible and cross-comparable research. ICEV task forces are a great resource for the latest information on standardization for different biofluids. Despite each biofluid's quirks, when it comes to EV isolation, size exclusion chromatography, or SEC, is commonly identified as one of the optimal methods across biofluids. SEC workflows, such as our QEV isolation platform, consistently deliver a good yield of high purity, high integrity EVs from biofluids. The QEV platform is scalable, automatable, and requires minimal hands-on time, making it a top choice for EV research. If you are looking to isolate EVs from a biofluid, our experts in EV isolation can offer advice to help you develop a protocol that gets the best out of your samples. We even have a service, PurePath for Diagnostics, tailored to developing the best EV isolation workflow to take you from raw biofluid to an isolate ready for your biomarker test. So if you're interested in biofluid EVs, reach out to our experts today.